What a great session, actually. It was really a lot of interesting talks and great talks. But now we're going to try to focus from nucleic acid to more models for cancer, such as organoids, single cells, and stem cells. So our first speaker for this session is Dr. Veronique Giroux, an assistant professor at University of Sherbrooke, and she would talk to us today about the organoid models and how to study stem cell biology in different digestive cancers.
So I hope everyone sees that now. Yes.
Yeah, that's great.
Awesome. Well, thanks so much, Samer, for the nice introduction and for the everyone for inviting me today. I'm really excited to be here. A little bit shy to be following such great experts in DNA repair, though. But I'll try to talk more about how we use organoid to study stem cell biology, and you'll see a little bit of DNA repair in there. So my lab mostly focused on the role of stem cells in GI diseases. We're studying as much normal stem cells and cancer stem cells, and we're in fact trying to use their really distinct function to be able to find new therapies for patients. By example, we know that stem cells have really high self-renewal as well as multipotent capacity. So that means being able to give rise to all different cell types from a tissue. So they're really important for tissue regeneration. So we're trying to see how we could act on these functions to be able to improve the regenerative capacities of patients that would have undergone inflammatory diseases or something like that. On the other end, we're also interested in cancer, where, and I'll come back to that later, cancer stem cells are highly involved in resistance to anti-cancer drugs. So we're trying to find a way to be able to find therapies that would be able to kill these cells and this way avoid cancer relapse, hopefully. So today I'll be focusing on the esophageal epithelium, one of the two main epithelia that I study in my lab. So the esophageal epithelium is a stratified squamous epithelium, which has a basal cell layer in which we will find all the proliferation. And these cells will undergo differentiation while they're migrating toward the lumen. So there's still a lot of controversy regarding if there is stem cells in the esophageal epithelium, which would reside in that basal cell layer. So there's still a lot of articles out there that still show different phenotype, in fact. Some of them show that this population would be heterogeneous, when some of them would say the opposite, so that it would be homogeneous. So that goes with two models that have been described for stem cells. The first one being the hierarchical model, the probably most known, in which it's really a pyramid, in which the stem cell is at the top, which have this self-renewing and multipotent capacities, and will be able to give rise to progenitor cells that we also call transit amplifying cells. These cells have high proliferative capacity, would be able to give this massive cell production on a daily basis to encounter for cell death, cell differentiation, or cells that would just be lost. And these cells then have the capacity to give rise to differentiated cells. There would be another model, which is called the stochastic model or equipotent model, in which it would be more an homogeneous population, in which all the cells would have the same capacity to either divide symmetrically for two stem cells, two differentiated cells, or asymmetrically, and then give rise to a differentiated cells and stem cells. So like I was mentioning, there was studies on both hands, and this is why I was studying that when I was still a postdoc fellow at Penn in Philadelphia. And this is when we discover or describe the first population of stem cells in the esophageal epithelium. I use for that K15 promoter. So this promoter encodes for cytokeratin 15, which has been described previously in the skin as being a stem cell marker for the stem cells in the hair follicle. So we were using mouse transgenic approaches to do lineage tracing. This is a gold standard technique in stem cell biology, which will allow us to label a specific cell type as well as all its progeny. So we use two different alleles. A first one that will drive an inducible Cre recombinase under the control of the keratin 15 promoter, so specifically in the K15 cells. And after we activate this Cre, we will induce a recombination and then a GFP expression, specifically in the K15 cells, and that will be transmitted to all the progeny cells. And when we do these experiments, we were able to identify the K15 cells and notice that with time, these cells are able to expand. So they have a clonal expansion capacity. They also have multipotency capacity. As you can see, they're able to give rise to the other cells of the esophageal epithelium. The normal K15 cells is here, and as it divides, it will form this kind of clone. And these cells are present up to 6 months that we've described. Here I show you a picture of 2 months, but the esophageal epithelium is renewed every week. So knowing that these cells are able to still maintain their proliferation way longer than that, supported that they have self-renewing capacity. And this is when I really got interested in organoid models, because organoid is one of the rare culture that will support the stem cell growth as well as maintenance, because it has this 3D matrix that will support that, as well as a media that comprise several growth factors that will be essential for their growth and to maintain this stem cell fate. So I developed organoid culture from normal esophageal cells. So these organoids will form a structure that really looks like an esophageal epithelium normally. So for this specific experiment, we sorted the K15 positive and K15 negative cells and put that in an organoid setting. And we could see that the K15 derived organoids were more capable to form organoids. So they have higher self-renewing capacity and clonogenic capacity. So that was a really good experiment that was able to support our hypothesis that K15 cells were in fact stem cells. And most importantly, when we look at the organoids that were derived from these two populations, we could see that they were morphologically really distinct. With the one derived from the K15 cells that looked really just like a normal esophagus, with the basal cells at the other layer, and the differentiated cells that were migrating toward the interior of the lumen. While the organoids derived from the K15 negative were all mixed up. It's like if they're not organized, the proliferation was restrained, the differentiation as well. So we continue to use that great model to study stem cell biology in my lab now. One last experiment I want to show you before I continue on stem cell is we study another capacity of stem cells, and this is resistance to radiation. So we use the lineage tracing approach, and following the induction of the GFP labeling, we irradiated the mice with a whole body radiation of a really high dose of 12 gray radiation, which would ablate most of the cells that are sensitive to radiation. And then we follow these mice in time. And we can notice in non-irradiated mice that we have this clonal expansion like we would expect. But following radiation, we could notice two things. First thing is that the K15 cells are still present, so suggesting that they are resistant to radiation, and also that they're still expanding, so suggesting that they're important in tissue regeneration following injury. And we've shown the same thing as well in the intestinal epithelia. So that was really key experiment that supported the description of the first esophageal stem cell in mouse, at least. So then we were really interested in what are really these cells? Are they also transcriptionally distinct from the other basal cells? And when we did RNA-seq, the first gene that was upregulated is MSH2, that everyone here would know is a mismatch repair gene. And we were able to confirm by Western blot that MSH2 expression is increased in K15 positive cells. So we undergo to study that in my lab, and our goal for that was to deplete MSH2 and to see if it was essential to maintain the self-renewal of the esophageal organoids. So as organoids are derived from primary cells, they're not really easy to transfect. So to be able to do some gene editing, the easier way is to go through lentiviral infection. So we first underwent to optimize a protocol to be able to infect these cells. And I'm not going to go through this. This is the recipe that we have in the lab, but what we need to understand is that an organoid arises from one single cells. So that will allow us that all the organoid has the same gene editing in this specific organoids, something that can be challenging for culture of organoids that will not arise from a single cell. So this is specific to the esophageal and makes that really easier. But we're now optimizing that in other type of organoid, and hopefully we'll get successful at that soon. So using that technique, we were able to use CRISPR-Cas9 to deplete MSH2 from these organoids. And we've done that really recently, but I thought for this specific people here, there might be interest for that. So I'm just going to show you the little data that we have so far. So we were able to confirm that there was depletion of MSH2. And when we look at Ki67 staining in these organoids, we could see already a diminution of the proliferative cells in the organoids depleted for MSH2. So supporting our hypothesis that this gene would be important for self-renewal and maintenance of the function of the organoid. When we derive organoid from these cells, we were able to measure as well their capacity to form organoid, and notice that by depleting SMSH2, this capacity was reduced. So supporting again the role of MSH2 in the self-renewal. We are now continuing these experiment and seeing if this role it's also playing in tissue regeneration, so response to radiation or response to chemotherapeutic drugs, but it's really promising. Like I mentioned at the beginning, stem cells are important in diseases, and unfortunately it's not a beneficial role, so it's more a detrimental role. So what we have noticed when we study esophageal cancer is that cancer stem cells have a high role in this. So for today, I'm going to focus on ESCC, standing for esophageal squamous cell carcinoma. This is the more prevalent esophageal cancer subtypes worldwide. So this is a disease that will arise from the esophageal epithelium due most likely to tobacco and alcohol usage, as well as thermal injury. So for people that drink a lot of hot tea or hot coffee, by example, it's a cancer that will be really prevalent in those type of populations. So coming back to stem cells, so we're talking about two different terms for stem cells in the context of cancer, and this is tumor-initiating cells and cancer stem cells. And most likely these cells are in fact the same, but they're not referring to the same function. So sometimes, not sometimes, but most of the people do a difference between those terms in the literature. So I just wanted to go over that a little bit to better understand what I'll be presenting later. So tumor-initiating cells would be that cells that have this capacity to form the tumor. So that will arise from a normal stem cell. So these normal stem cells have high DNA repair capacity. Since they're proliferating a lot, they need to be able to repair their DNA really efficiently, so they're not transmitting mutation. Even that, they will still acquire mutation, unfortunately. And when they acquire sufficient number of mutation, then they would become a tumor-initiating cells and be able to form a tumor. Cancer stem cells would more refer to that subpopulation of cells in a tumor that have high self-renewing capacity, so be the main driver toward the tumor growth, but also higher multipotent capacity. So that would refer more to tumor heterogeneity. So why in a same tumor we would have different clones, mutation clone, but also functional clones. And lastly, another function that cancer stem cells have been associated with is resistant to anti-cancer therapies, such as chemo and radiotherapy. And this is one of the aspects that we're specifically interested in the lab. Okay. So the first thing we did is to see if K15 cells have a role to play in esophageal carcinogenesis. So we use a foreign QO model, which will mimic the ESCC disease that we see in human. So this is a carcinogen that will be given in the water of the mice. And this mice, after several weeks though, will develop a tumor in the esophagus as well as in the oral mucosa. When we look histologically, we will see different type of lesion. So hyperplastic lesion as well as full-blown tumor lesion. So what we did is we combined the lineage tracing approach with this type of model. So we label with GFP the K15 cells, and after did this foreign QO model, which is spread on almost 6 months. And then we look at the GFP clone. And we could see that in an ESCC setting, the clones derived from the K15 were less in number, but they were really bigger. So supporting that K15 growth or proliferation is higher in the context of esophageal carcinogenesis. And this is just a counting of these clones. So that was supporting that K15 cells might have some tumor initiating capacity. To study the other end, so more the cancer stem cell approach, we combine those two approaches again, but in a different time manner. So we did a foreign QO treatment, but induced the GFP labeling only at the end of this treatment. So that would allow us at this time to label the K15 cells that are already in the tumor and not the one that will give rise to the tumor. And then when we look at this esophagus, so this is an open esophagus and we're looking upward, and you can see that there's a GFP lesion, like you can see right here. And there's also part of it that is still K15 negative, and some of them look even like lesion. So most likely there's other cells that are able to acquire this type of capacity, or it's not all the cells that are still K15 active, right? Because we can see tumor here that have a little part GFP could be the one that originated the tumor. But when we did the labeling, there are still cells that were not active at that time. And what we did is that we use flow cytometry to sort those two populations, and we derive organoid from that to study more tumorids, so an organoid that is derived from cancer tissue. And really interestingly, we notice strong morphological changes in the organoid derived from these two cell population in a tumor setting. So we can see that the organoids derived from a K15 cell population is less virid. So normally that would suggest that there's hyperplasia. So all those protrusion that are growing outward would be clones of cells that are highly proliferative and that will form those kind of buds. Contrarily to a more spheric organoid that looked like a normal organoid in reality, which probably the growth was really more controlled. So that suggests that the K15 population probably have higher proliferation capacity and tumorigenic capacity. But this would still need to be reimplanted in mice to confirm that they are more tumorigenic. That being said, though, we did not observe that phenotype in all the organoids from a K15 population. It's a really mixed phenotype. So the other phenotype we observe is the structures look more spheric for both, but the one that are derived from the K15 have different expression of differentiation and proliferative markers. So we look at K13, which is a differentiation marker in the esophagus, as well as P63, a member of the P53 family. But in our world, it will stand for the basal cells, so the cells that are the most proliferative. And when we look at that, this is more what we would expect. And this would be an organoid that is not prone to differentiation. So that recapitulates more what we had observed from normal esophageal epithelium. Why we see those two phenotypes? It's probably because we were taking the entire esophagus. So there's not necessarily lesion everywhere. So that was probably more from normal esophagus or less tumorigenic. And this is what probably coming from really full-blown tumor. So now what we're trying to do, because remember that an esophagus in a mouse is quite tiny, so we're trying to micro dissect the tumor to be able to isolate the K15 cells from the tumor and from the normal and compare all of that. And I've not put the results here because it's really preliminary, but it looks really exciting so far. From the tumor, if we sort only these cells, it looks like only K15 positive cells are able to form organoid. K15 negative are able to do it at first, but as soon as we passage them, they're not able to grow. So suggesting that the stem cell pool would really be only in the K15 positive population, but we still have to repeat that. But this is a really exciting experiment that we just got. So coming back to this cancer stem cell phenotype, there's different markers that we can look to use we can use to look at cancer stem cell population, and two of them is CD44 and CD24. So we did flow cytometry analysis on those two different organoid population, and we could see that K15 derived organoids have a higher proportion of CD44, CD24 I cells, suggesting a higher proportion of cancer stem cells. So that was going again with our hypothesis. And now what we're trying to do is to see if these cells will also be more resistant to anti-cancer drug like we would expect. And if that's the case, then we want to find new drugs that would be able to kill these cells. So going most likely with a high throughput drug screening approach. So what I've shown you today is that organoids are a great model. I think it's one of the best, in fact, personally, but they can be used to do a lot of different things. What I showed you mostly today is that it can be used to model diseases, but it has been used for variable different application, like stem cell biology more in the context of development, regenerative medicine to form kind of tissue that could then be reimplanted in patients. Drug screening is really a nice approach that we can do in this type of culture as well. So we have used that technique to study stem cell biology in normal and tumor context. And in normal setting, we have used that to show that K15 cells are clonogenic, that they are resistant to high dose radiation. And we're now studying the role of MSH2 in these organoids. And we've also seen increase in other gene associated with DNA repair, like RIDE51. So we're now trying to establish CRISPR-Cas9 for this in organoid, but it has been a little bit more challenging than for MSH2 so far. And in the tumor, I think this is where it worked the best because we saw a lot of morphological changes, and that makes our life way easier because it's so easy to describe these phenotype. So it's been really exciting to use this model in our research so far. So with that, I would like to thank the people in the lab that does most of the work. So especially Julie and Marjorie, two grad students that have been working on this project, as well as Dominique Jean, a research assistant. I would also like to thank all the funding sources that we have. And with that, I'll welcome any question that you could have.